Okay, this is video 1302, and this is section 13.2 in your textbook, and this is on translation. So the first step in decoding genetic message is to transcribe a base sequence from DNA to RNA. And remember, a little section of DNA, I've said it over and over, is considered a gene. Now this little base sequence is going to code for a certain protein. And if you remember, let's review a little bit. Proteins are made by what monomer? That's right, amino acids, and together they form long chains called polypeptides. Okay? And there are as many as 20 different amino acids that are commonly found, and usually we get them from the food that we eat, in particular proteins that we eat. Now, this genetic code is found or comes about due to specific amino acids and a specific sequence of these amino acids. And this sequence influences the shape of the protein, and the shape of the protein determines the function. It also determines the properties of the proteins. If you remember, RNA has 4 different bases: adenine, cytosine, guanine, and uracil. These letters, or these A, C, G, and U, form what's called your genetic code. Okay, so being that there's just 4 letters, okay, the codon actually consists of 3 letters. Okay, so 3 letters make what's called a codon, and that's what's going to code for a certain amino acid. This right here is what you're going to be using to determine the genetic code, or to determine the amino acids that are going to be in a protein. So for example, I said earlier there's 20 different amino acids. Well, since there's only there's 3 to a codon, right? That means that more than one codon can code for the same amino acid. More than one codon can specify the same amino acid. So if you look at leucine here, UUA, UUG. Where's UUA? Here's U, and you go to A, that gives you leucine. UUG gives you leucine. CUU, CUU gives you leucine. CUC, CUA, and CUG also give you leucine. Now, this genetic code that we're talking about also has punctuation marks. So there's a special codon that's used for the protein to start being made. And the start codon is actually AUG. Now AUG, I always remember this one because school starts in August, and that's the start codon to start making the protein. So if you look at this, let's look and see what the stop codons are. So here's one stop codon, UAA, UAG. See if you look at that, right? There's another stop one here, UGA. So University of Georgia is also a stop codon. So using this wheel for the genetic code, you need to determine what the amino acids are. So AUG, so you go A, you stay in that quadrant, A, U, G, methionine, which is also, remember, that's our start codon. GAC, GAC. Usually just write the first 3 letters of the amino acid. AAG, lysine. GGA, glycine. And UGA, that was stop, remember? So see if you can stop the video and go ahead and do these. What's the amino acid for UGG? UGG. That's right, tryptophan. So what happens is ribosomes use the sequence of codons in the messenger RNA to assemble these amino acids into polypeptide chains. This is called translation. The decoding of a messenger RNA message into a protein. So this right here would be our messenger RNA. See, this is our messenger RNA, and it goes to, this would be the ribosome. And these right here are transfer RNAs. Do you remember what transfer RNAs do? That's right, they carry the amino acids. So in translation, the messenger RNA is transcribed in the nucleus. Okay, so we get that our messenger RNA, and then it leaves the nucleus through the pores in the nuclear envelope, and then it goes to the cytoplasm for translation. And so here are the steps. So the ribosome will attach to the messenger RNA. So remember our ribosome. Remember we had ribosomal RNA in it. The ribosome will read each codon, so each group of 3 letters. Then the ribosome will direct the transfer RNA to bring the specific amino acid to the ribosome. The way it does this is that on the transfer RNA, this is a transfer RNA, it has to match up with the codon that's on the messenger RNA. Okay, so you see AAA matches up with UUU. And then as it does that, it will attach the amino acid. The ribosome will attach the amino acid to the polypeptide chain. So the next one coming in would be this lysine. So that one eventually would get attached to it. Then when it hits the stop codon, the ribosome will release the peptide chain. So each tRNA carries only one kind of amino acid, and it has what's called the anticodon. So that was what this would be. But all we worry about is the codon, because that's what we use that little wheel on. Excuse me. AAA is R. So the transfer RNA holds the ribosomal proteins in place, and it locates the beginning of the messenger RNA message. It can also carry out other chemical reactions that help to join the amino acids together. Do you remember what the central dogma is? That's right. DNA codes for RNA, which guides protein synthesis. Another way to look at that would be that hereditary information, that's the DNA, right, is transferred from DNA, right, to RNA. That's called transcription, and then from RNA to protein. That's called translation. Here's another way to look at it. So DNA replicates, copies itself. It transcribes it for RNA synthesis. So we got DNA replication, RNA synthesis. That RNA leaves the nucleus, goes to the cytoplasm where the ribosome is, and then we have protein synthesis through translation. So most genes have constructions for assembling proteins. So if you get a gene, remember it's a piece of DNA, that's the instructions for a certain protein. Now, we know that proteins are enzymes, which makes them very important because they catalyze and regulate chemical reactions. Here's some examples of things that proteins do. These are some other phenotypes that they do. Produce pigment, regulate patterns of tissue growth, trigger the female or male pattern of development. Proteins are microscopic tools that are specifically designed to build or operate a component of a cell. And gene expression is the way in which DNA, RNA, and proteins are involved in putting this genetic information into action. So DNA carries the information, okay, and these sequence of bases are the template for making the messenger RNA. Those codons, those groups of 3 bases, specify the sequence of amino acids. Proteins, which are made from those amino acids, play a key role in producing an organism's traits. So with the genetic code, remember that it's nearly universal. So for all living organisms, they use the same bases. They are always read 3 bases at a time. That's your codon, and it always goes in the same direction. So if you think about the enormous diversity of living organisms, it's remarkable that at the molecular level, they are still read 3 bases at a time and in the same direction, and they all have the same bases in them, in their RNA.